In my laboratory at the University of Sussex, we are working on a broad field of learning and memory research, and in particular, we are interested in the cellular and molecular backgrounds underlying learning. Now, it is very difficult to study these processes, especially on that level, on the cellular and molecular level, in the human brain. So we are using the brain of the pond snail, Lymnaea stagnalis. Its brain is much simpler than the very complex human or mammalian brain, and it has huge neurons, and these neurons are very well identified, so we know the circuits which are activated by specific learning. Surprisingly, these snails can learn very quickly and remember for a very long time. So we can actually look at the formation of the memory, and we are especially interested in the first bit, which is called memory consolidation, which means that once you learn something, then there is a time while the memory gets stronger and gets resistance to interference.
So I'm using intracellular electrophysiology to record from identified neurons within the snail's central nervous system. And we can record this activity and we see what changes occur after the animal has learned a new association or it's undergone some sort of change in its motivational state. For example, if the animal's hungry or it's recently eaten, how does activity in the brain encode this information, and what changes cause alterations in the animal's behaviour? We use a multitude of techniques. So we record the animal's behaviour and we see what happens in the animal's behaviour, and then we look down to the level of individual neurons and see how activity changes using sharp electrodes, so intracellular electrophysiology. Here we can see intracellular recordings from a feeding interneuron and a motor neuron in pink and purple. We're also recording extracellularly from the muscles which produce the feeding behaviour. And this sequence of events underlies the feeding behaviour which we observe behaviourally. And using this rhythm, we can extrapolate changes which occur in the brain which then underlie these behavioural changes that we are observing after the animal has learnt an association.
You can train the snails to associate a chemical stimulus with food, and we use this ability of the snails to look at what happens during the learning process. We can time either the interference or the memory performance at very sharply timed points after the training. So this allowed us to discover that this process of consolidation is not a simple progressive process, but there are lapses when the memory, when it is more difficult to recall the memory.
Any learning that affects the brain will leave a molecular trace, a biochemical trace in the neurons, and therefore it is important to discover what these molecular changes are. The molecules we are looking at are exactly the same that are being activated by learning in the human brain. And the principle of the work in the histology lab is that you take tissue from these brains, animal brains, and then you use specific assays such as immunohistochemistry or in situ hybridization to look at learning-induced changes in the amount or the activation level of these molecules. But in this laboratory we are doing a bit more than that, because we are also using biochemical methods and molecular methods to look at dynamic changes induced in the activity level of these molecules by learning.
We're looking at the ganglia of snails, and we're looking for particular proteins within the neurons which we've stained using antibodies, and it gives a very good idea or indication of the location of many of the proteins at different stages of learning. The green is showing high levels of Krebs expression, or we know is associated with memory and learning. The red is showing serotonin, the serotonin network in the ganglia, and the blue highlights the nuclei of all the neurons within the brain. Where biochemical techniques give us an indication of the levels of protein expression within the brain, visualizing the brain using confocal techniques allows us to see specifically what neurons are expressing these proteins in larger quantities during the memory process.
These memory lapses during the consolidation are periods when it is much easier to interfere with the memory trace. And this has also clinical applications, because there are conditions like the post-traumatic stress disorder, when people go through a traumatic event, and then the treatment is that they try to kind of make these events, make the people forget these events. So now, based on our research, we know that during these periods, so there are specific time points when it is actually much more successful to interfere with the memory. And that's what our latest research is about. We are trying to induce actually new memories during these time points, so not just erase the original memory, but replace it with something which hopefully more pleasant or more better for the individuals. And we're trying to do it on the snail.